Afternoon, everyone. Thanks for being here. I'm Sabrina, and this is Remi. Today, we will be presenting our final briefing on the characterization of IgGShredder, a novel antibody digesting enzyme. I will cover the project context, and the key results, and then Remi will continue with the summary and recommendations. In this study, we use purified rabbit anti-ship red blood cell IgG as a model antibody substrate and compare undigested IgG, IDZ digested IgG, and IgGshredder digested IgG. Everything we are showing today is based on our own process experimental data, and we have presented it as a true reflection of what we obtained. Antibody digesting enzymes are very important because where IgG is cleaved determine which structural domain are retained, and that directly affects functions such as antigen cross-linking and Fc-dependent effector activity. This is highly relevant when we want to generate antibody fragment for therapeutic use. So our aim was to determine how IgGshredder cleaves IgG, compare its overall digestion pattern with IDZ, and assess how digestion changes antibody structure and also function. To address that aim, we used three complementary assays. First, hemagglutination assay was used to see whether digestion changed the antibody ability to cross-link ship red blood cell. So this mainly tells us about retained Fab-mediated binding valency. Second, we used reducing and non-reducing SDS page to look at fragment size and composition, so we could link the structural changes back to the functional result. Finally, we use a phagocytosis assay to assess whether digestion alters Fc-dependent opsonization and also uptake. We first assess whether each antibody preparation could still cross-link ship red blood cell. As shown on the plate and in the titer table, the undigested showed the highest hemagglutination titer, which was 8. The IDZ digested sample retained partial hemagglutination with a titer of 4. In contrast, the IgGshredder digest retained only minimal hemagglutination activity with a titer of 1. So compared with IDZ, IgGshredder caused a much greater loss of ship red blood cell cross-linking. Under non-reducing condition, where interchain disulfide bonds are preserved, the undigested IgG migrated as a single band at around 150 kilodalton, which is what we would expect for intact antibody. The IDZ digest show band at approximately 86 and 42 kilodaltons. The IgGshredder digested sample was dominated by a 42 kilodalton species and did not show the larger 86 kilodalton band seen in the IDZ sample. Under reducing condition, where the disulfide bonds are broken, the undigested IgG show band at approximately 56 and 26 kilodaltons, consistent with heavy and light chain associated species. The IDZ digest show a dominant 26 kilodalton band. The IgGshredder digested sample show a 26 kilodalton band plus an additional smaller band at around 14 kilodalton. Together with the non-reducing gel, this suggests that the IgGshredder digest underwent further fragmentation and did not behave like the IDZ digest. Finally, we assess Fc-dependent function using a phagocytosis assay with fluorescently labeled ship red blood cell and raw macrophages. In this experiment, the ship red blood cell only sample act as the negative control and gave 17.5% cell bright green positive raw cells, representing the baseline uptake without antibody opsonization. The undigested anti-ship red blood cell IgG act as the positive control and increased uptake to 47.9%, which is consistent with intact Fc-mediated opsonization. The IDZ digest reduced uptake to 20.6%, which was close to the baseline. The IgGshredder, on the other hand, gave 30.5%, so uptake was clearly reduced compared with the undigested sample, but not completely lost. I'll now hand over to Remi, who will integrate this finding and present our summary and also recommendation.
This figure shows our predictive cleavage pattern. This suggests that IgGshredder appears most consistent with papain-like cleavage. The reduction in hemagglutination suggests that the Fab region is not intact. In bivalent form, we expect increased cross-linking activity similar to IDES, which produces intact Fab2. Therefore, IgGshredder's low hemagglutination supports cleavage in an upper hinge region, resulting in separated Fab, like papain. This is supported by the non-reducing SDS page. Papain-like cleavage is expected to produce fragments of approximately 50 kilodalton, which is consistent with the single major band observed for IgGshredder. In contrast, IDES digestion produces two bands, an Fab2 fragment at 100 and an Fc at 50 kilodalton. This further supports separated FAB of IgG-shredder cleavage, as no higher band was observed. Under reducing conditions, disulfide bonds break, so hinge cleaved fragments should show smaller bands. Both IDES and IgG-shredder showed bands at approximately 25 kilodalton, supporting hinge cleaving. The phagocytosis assay supports further as IgG-shredder showed reduced phagocytosis compared to undigested IgG, indicating Fc-mediated opsonization was affected. This suggests that IgG-shredder reduces Fc-dependent phagocytosis, consistent with papain-like cleavage where Fc is separated from FAB. Unexpected results. Firstly, a low molecular weight band at approximately 50 kilodalton on the reducing gel was unexpected and did not align with the non-reducing gel, as no corresponding fragment was seen. If the band was a cleavage product from FAB or FC region, a band should appear in the presence or absence of disulfide bonds. It is possible the low molecular weight cleavage product was too faint to see on the non-reducing gel, was poorly resolved, migrated too close to the dye front, or was below the stain detection limit. If not, it could have been an experimental artifact, such as minor sample degradation, uneven loading, or unimportant products of digestion. To rectify, repeat the digestion to confirm whether the band is reproducible and use a higher percentage gel for smaller protein. If relevant, the exact origin of the small band cannot be determined from the data alone, so it was not incorporated in the IgG-shredder cleavage prediction. Secondly, the phagocytosis assay. Although IgG-shredder reduced Fc-dependent function, phagocytosis level was higher than baseline and IDES, so some Fc-mediated activity occurred. While this can suggest IgG-shredder digested IgG has partial preservation of Fc function, it's unlikely the main explanation. Digestion may have been incomplete and proportion of IgG may have remained in the sample, as typical enzyme digestion protocols require digestion optimization by assessing a range of incubation times. Our antibodies were incubated for 1 hour, which may not have been sufficient for complete digestion. To rectify, either use pre-activated enzyme and incubate for minimum 6 hours or perform a digestion time curve using STS page for digestion percent. Either Fc function preservation or residual intact IgG cannot be confirmed from the data. A therapeutic application of antibody fragments is toxin neutralization, such as antivenoms like Crofap. Fragmented antibodies' smaller size allows rapid diffusion through blood and tissues, enabling faster toxin neutralization. In addition, absence of the Fc region reduces the risk of excessive immune activation and serum sickness for antitoxins. IDES's intact FAB2 is larger, can cross-link toxins into immune complexes, but Fc function is removed, so the complexes are cleared less efficiently, so they may linger in the circulation. IgG-shredder has low cross-linking and smaller fragments that diffuse more rapidly, so may neutralize without forming complexes. Therefore, IgG-shredder is a better antitoxin, as Fc-mediated mechanisms are reduced and fast neutralization is prioritized over toxin clearance via immune complexes. A real-life example is botulism toxin, where antibodies could prevent the cleavage of the toxin, keeping it in its unactive form. And here's a very simplified diagram of it. IgG-shredder could work better here compared to IDES or pepsin, as typical treatments require low immunogenic designs. For a sales pitch to a consumer, IgG-shredder offers advantages over other enzymes, as its fragments are smaller compared to IDES and pepsins for faster neutralization of the target and has low cross-linking unlike IDES and pepsin, which is preferred if generating hard-to-clear immune complexes is unwanted. Thank you.